Imagine for a moment a medicine, maybe one for an autoimmune disease, that truly transforms life for one person, offering significant relief,
Hmm.
yet for someone else, it just falls flat. Or maybe even worse, triggers unexpected side effects.
Yeah, that happens.
What if the crucial difference wasn't really about their diet or even their specific diagnosis, but something, well, something as fundamental as their ancestral background?
It's a profound thought, isn't it? It really points to these invisible variations within us, variations that biomedical research has for too long, kind of inadvertently overlooked.
And that oversight is precisely what we're tackling in today's deep dive. We're exploring the Asian Immune Diversity Atlas, or AIDA.
AIDA, right.
This is a truly significant study because it shines a light directly on what seems like a massive blind spot in medical science.
You're absolutely right. The core issue, historically, has been a severe lack of diversity in our foundational genetic and genomic data sets.
Right, the basic building blocks.
Exactly. These critical resources, things like the GWS catalog or the GTEx project, they've been overwhelmingly skewed. We're talking about roughly 86% of the data coming from individuals of European ancestry.
86%.
Yeah, even though that group makes up only about, 15% of the world's population?
That's just a staggering disparity. It means our collective understanding of, well, everything from disease risk and drug responses to effective diagnostics is just incomplete.
It is. And this isn't just some abstract problem for researchers in a lab. It directly contributes to health disparities globally.
How so?
Well, think about it. A molecular diagnostic test that's, spot on for one population might entirely miss the mark for another.
Okay.
And that could potentially delay treatment or even lead to a misdiagnosis.
Which brings us right to the crucial role of this AIDA project.
Exactly. It steps right into this gap by building a multinational single-cell reference atlas.
Okay, single cell reference atlas, break that down for us.
Think of it like mapping the immune system's really intricate landscape. They do this in healthy individuals across diverse Asian populations, but, and this is key, with an unprecedented level of resolution.
Okay.
And what's particularly powerful here is that AIDA isn't just looking at broad continental differences, like Asian versus European. It really emphasizes the subcontinental human diversity present within Asia itself.
Oh, so much more granular.
Not anymore.
So as we unravel this today, you'll discover just how profoundly factors like age, sex, self-reported ethnicity, and specific genetic variants can actually shape our immune cells. And this isn't merely fascinating biology, it's absolutely vital for developing precision medicine that, actually works for everyone, no matter their background.
So after setting the stage with that critical need for diversity, let's zero in on AIDA itself. What exactly is this benchmark project, and how did they achieve this sort of unprecedented level of detail?
Okay. So at its core, AIDA is a massive single-cell RNA sequencing, or cRNA-seq, healthy reference.
It's cRNA-seq.
Right.
Yeah. For those maybe not familiar, cRNA-seq is kind of like taking an incredibly detailed photograph of millions of individual immune cells.
Okay, individual photos, not a group shot.
Exactly. Instead of that blurry group shot that averages everything out, you get a crystal clear image of each cell, capturing its unique genetic activity, basically what it's doing at that moment.
Ah, okay.
This allows for a granularity, a level of insight that was frankly impossible before.
That's a really helpful analogy. And the sheer scale of this project is quite impressive, isn't it? Just the numbers involved.
It absolutely is. The atlas comprises 1,265,624 circulating immune cells,
Wow.
collected from 619 healthy donors.
Over 1.2 million individual photographs. And the reach is equally impressive, ensuring that broad representation you mentioned.
Indeed. These donors span 7 distinct population groups across 5 Asian countries. So you have India, Japan, South Korea, Thailand. And then in Singapore, they specifically included individuals of Chinese, Malay, and Indian ethnicities. So you see, it's this broad geographical and ethnic inclusion that's really crucial for capturing the true diversity within Asia.
And I gather it wasn't just about collecting a lot of data, right? The rigor behind this study seems to be a real highlight.
Precisely. That's critical. The researchers meticulously controlled for technical variations across all those different study sites.
How did they manage that? That sounds complicated.
It is. They harmonized every step,
Mm.
donor selection criteria, how the samples were processed, the experimental protocols, everything was standardized.
Okay.
They even employed something called genetic multiplexing for the samples.
Genetic multiplexing.
Yeah, a pretty sophisticated technique where you tag samples genetically, which allows them to be processed together in the same batch, and then you can sort of deconvolute them computationally afterwards.
Oh, clever.
It really minimizes those technical confounders, things that could introduce noise. It helps ensure that the differences you observe are genuinely biological, not just, artifacts of slightly different lab procedures.
That meticulous approach really underlines why the single-cell resolution is so powerful. You mentioned traditional studies often look at bulk tissue,
Right, the average.
which is like averaging all those individual photos, but say-RNA-seq gives us this incredibly granular view. It lets us see how individual cells behave and differ, right? Even the really rare ones.
Exactly. Even rare subtypes like, say, DNT cells, which might only make up 0.04% of all cells, or CDC1 cells, also around 0.04%, and atypical B cells maybe at 0.4%.
Tiny population.
Tiny, but potentially very important. These are highly specialized immune players, often totally missed or averaged out in bulk studies. Seeing their unique behaviors unlocks a much deeper understanding of our immune system.
It really provides a kind of blueprint for understanding the subtle nuances, doesn't it?
It does. This level of detail allows the identification of variations, maybe even novel cell populations, things like SCARD1 high ILC and XCL1 high ILC that they mentioned, they would just be completely obscured in bulk analyses.
It's like finding unique specialized roles within an orchestra, maybe, each contributing in its own distinct way.
That's a great analogy, yeah.
So for you listening, think about this. What do you think seeing these individual cell details, rather than just averages, allows us to understand about the immune system that was previously hidden? And why is that so powerful for our health? Okay, so just to recap where we are, about 5 minutes in, we've unpacked the AIDA project, this monumental effort to map immune diversity, what it is, a meticulously built single-cell RNA sequencing atlas, its scale, over 1.2 million immune cells from 619 healthy donors,
Huge numbers.
its reach, spanning 7 diverse Asian population groups across 5 countries, and its power. That single-cell resolution uncovers the granular behavior of even rare immune cell subtypes. Okay. So, with that incredible detail on how AIDA was built, the real question is, what did it actually uncover about how our individual identities, who we are profoundly, shape our immune system?
Well, what's particularly striking, I think, is that AIDA revealed self-reported ethnicity, genetic ancestry, and sex had comparable impacts on immune cell properties.
Incomparable to what?
Comparable to each other, but often more significant impacts than factors we often focus on, like age, BMI, or even smoking status.
Wow, okay. So ethnicity, ancestry, sex right up there. Maybe even more important than age or lifestyle factors in some respects.
In terms of explaining the variance in certain immune cell features, yes. It immediately flags them as really fundamental influences we absolutely must consider.
That is a significant finding. It definitely challenges some common assumptions. Let's maybe delve into age and sex first. What did AIDA tell us there?
Sure. So the atlas confirms some known patterns. For example, plasmacytoid dendritic cells, or PDCs. These are important antiviral cells. Their proportions decreased with age. That wasn't entirely unexpected.
Okay.
In terms of sex, B cells were found to be more abundant in females, while natural killer, or NK, cell proportions were higher in males. Again, some previous hints of this.
Right.
But AIDA also provided more refined insights. For instance, CD8 plus T naive cells. These are sort of inexperienced killer T cells. They were uniformly depleted in individuals 50 years and older compared to younger donors.
A clear age drop-off.
Yes. But then, here's where it gets really interesting. CD4 plus key cytotoxic cell proportions. These are helper T cells that gain killing functions. They actually increase with age.
Increase? That's counterintuitive.
It is. And they were even found to be heightened in supercentenarians, people living over 110 years old.
Whoa, so what does that suggest?
Well, it suggests a potential role for these specific cells in extreme longevity. Perhaps they adapt somehow to maintain immune function late in life.
That's fascinating. Seeing some cells decline while others actually increase with age, especially in those living to exceptional ages. Okay, now let's talk about self-reported ethnicity. The findings here sound like they offer some truly eye-opening insights into these population-specific differences.
They absolutely do. They're quite stark in some cases. For instance, Singaporean Malay donors, referred to as SG Malay, in this study showed elevated B cell proportions. They also had an enrichment of gamma delta T cells, or GDT cells, another specialized T cell type.
Okay, different levels there.
Then you look at Thai donors. They had lower myeloid cell proportions overall, and specifically lower monocyte proportions. And this wasn't just in the single-cell data. It was independently corroborated by standard complete blood count data.
Ah, so multiple lines of evidence.
Exactly. And that tells us something really practical. A universal normal range for these common blood cell counts simply isn't sufficient. Ethnicity clearly needs to be factored into our diagnostic baselines.
So if you're a Thai patient, your normal might look quite different from someone else's normal, and that needs to be recognized in a clinical setting, right?
Precisely. It has direct clinical relevance.
Mm-hmm.
Then, Singaporean Indian donors, or Eskindian, for example, they had lower natural killer NK cell proportions compared to others.
Okay.
And perhaps one of the most notable findings was in Korean donors. They showed significantly lower T cell proportions overall, and strikingly, much lower regulatory T cell proportions.
Yeah.
TREGs.
Lower TREGs. That sounds significant. You hear a lot about TREGs and autoimmunity. What are the potential implications there? Maybe for susceptibility to autoimmune conditions or even just the overall immune balance in that population?
That's an excellent question, and it's exactly what researchers would ask next. You can imagine TREG cells as maybe the immune system's peacekeepers or traffic cops.
Okay, keeping things in check.
Right. Their main job is to prevent the immune system from overreacting and mistakenly attacking the body's own tissues, which is, the hallmark of autoimmune diseases. So having lower numbers could potentially imply a differing immune regulatory balance. It might influence susceptibility to certain autoimmune conditions, or maybe even affect how those conditions present clinically in the Korean population compared to others. It opens up a whole line of inquiry.
That analogy really helps clarify their role, and the study also found links for MAI T cells, mucosal-associated invariant T cells.
Yes, their proportions were significantly correlated with genetic ancestry, specifically looking at genotype PC1, which captures a major axis of genetic variation among the Singapore donors. It just reinforces that these links between ancestry and immune cell composition run deep.
And it wasn't just single factors, right? Did they see interactions between age, sex, and ethnicity?
They did, adding another layer of complexity, which is, of course, how biology really works. For instance, CD16 plus NK cells, a major subset of NK cells, are typically higher in males. That's a fairly well-known sex difference.
Okay.
But this effect was not observed in the Thai donors in this study.
Huh. So the sex difference wasn't universal across populations.
Apparently not in this cohort, which raises fascinating possibilities, maybe about population-specific sex hormone activity, or perhaps unique gene regulation differences at play in Thai individuals affecting those NK cells.
Interesting. Any other interactions?
Yes, another clear one was with age. Korean and SG Malay donors showed sharper decreases in their CD4 plus T naive cell proportions with age compared to SG Chinese donors.
So aging affects these cells differently depending on your background.
Exactly. It clearly underscores the need for not just age-specific, but age and population-specific reference ranges in clinical diagnostics. You can't just use one or the other. You likely need both.
So thinking about this for you, the listener, if your doctor relies on normal ranges for immune cell counts, and those ranges are based on, say, a predominantly European population, how might that affect your diagnosis, your treatment, or even just how your personal health is understood if you have a different ancestry? Yeah. It really highlights the urgent need for more tailored approaches in medicine. Okay, let's do a quick recap around the 10-minute mark. AIDA has powerfully demonstrated how our individual human diversity, our ancestry, our age, our sex, profoundly shapes the very makeup of our immune cells.
Clear signals.
Key influencers. We saw that ethnicity, ancestry, and sex have impacts comparable to, and sometimes even greater than, age on immune cell features, age-sex specifics. Age affects PDCs and CD8 plus T-naive cells, but can even boost CD4 plus T-cytotoxic cells in the very elderly. B cells are higher in females, and K cells in males, generally speaking.
Generally, yeah.
Yeah. Ethnic differences. Singaporean Malay, Thai, Singaporean Indian, and Korean populations all show distinct patterns in various cell types like B cells, myeloid cells, NK cells, and notably, those regulatory T cells interactions. And crucially, these factors interact. Sex differences in NK cells aren't universal, and age-related cell declines vary by ethnicity.
The complexity is key.
These aren't just, academic curiosities. They have direct, real-world implications for how diseases are diagnosed and understood. Now let's dig even deeper. Let's get into the molecular blueprints, exploring the genes, their expression levels, and how they might link more directly to disease.
Absolutely. Because AIDA didn't just stop at counting cell proportions. They used advanced computational tools, like one called MELOR, to examine what they termed cell neighborhoods.
Cell neighborhoods, like little communities.
Essentially, yes. Groups of cells that have highly similar gene expression patterns, even within the same broad cell type. This lets you find even finer grained differences.
Okay, so zooming in even further.
Exactly. For example, in older donors, they found specific neighborhoods within the CD16 plus NK cell population. These neighborhoods had unique gene expression signatures. They mentioned FCR1 GLOW and KLRC2C, and these specific neighborhoods were enriched in the older group.
And what does that suggest?
It suggests an age bias in what we call adaptive NK cells. These are NK cells thought to gain some memory-like functions, perhaps after encountering viruses. So aging might specifically boost these experienced NK cells.
That's a powerful refinement, moving beyond just broad cell types like NK cell to look at specific communities of cells with shared characteristics, shared jobs almost.
Precisely. And they also pinpointed a neighborhood within the CD4 plus T naive cells, characterized by heightened expression of a gene called SOX4. This particular neighborhood was the most depleted in older donors.
Ah, so of a very specific decline.
Yes, it gives us a much more refined molecular signature of T-cell aging than just saying naive T-cells decline. It offers a clearer molecular picture of how our immune system changes over time.
Beyond these neighborhoods, you mentioned AIDA identified thousands of differentially expressed genes, DEGs.
That's right, DEGs. These are genes whose activity levels, their expression, varied significantly across the different population groups, but within specific immune cell subtypes. So comparing, say, monocytes in one group versus another.
Okay. Can you give an example?
Sure. A gene called UTS2. It's known to be involved in vasoconstriction, narrowing blood vessels, and has links to metabolic traits. This gene was found to be differentially expressed in 16 different immune subtypes across the various populations studied.
16 subtypes, that's widespread.
It is. And this discovery strongly points towards potential population-specific differences in metabolic health or maybe cardiovascular function that are reflected in the immune system and really warrant further investigation.
And this is where we start seeing the genetic roots of some of these differences more clearly, right? Like with the FCR1A gene.
Exactly. FCR1A is a gene implicated in the risk for allergic diseases. What they found was that this gene was upregulated, more active in Singaporean and Indian donors, but downregulated, less active in Singaporean Chinese donors, specifically in their CD14 plus monocytes.
And is there a reason for that difference?
It appears to be linked, at least partly, to variations in a specific genetic variant nearby, called RS242787. The different versions or alleles of this variant have different frequencies across these ethnic groups, so the underlying genetic difference likely drives the difference in gene expression.
Wow, so that's a perfect illustration, isn't it? A subtle genetic variation, common in one population but maybe less so in another, leads to measurable molecular differences, like how active a gene is, which in turn could influence things like allergy risk.
Precisely. And similarly, they found that in Singaporean Malay CD14 plus monocytes, there was an upregulation of a whole set of genes associated with inflammatory responses and host pathogen defense. This echoed what they saw in certain enriched cell neighborhoods that had high expression of interferon-associated genes. It suggests perhaps a sort of baseline primed or more alert immune state in the monocytes of this group.
And it's not just about which genes are active, but also how they're regulated. AIDA looked into gene regulatory networks too, regulins.
Yes, absolutely. Regulins are networks where one master regulator gene controls the activity of many other target genes. They found differences here too. For instance, the activity of the ZBTB7A regulin.
Okay.
Its activity in Treg cells, those regulatory T cells we talked about, was heightened in male versus female Singaporean Indian donors. But this sex difference wasn't seen in the other Singaporean ethnicities, the Chinese or Malay groups.
So a sex difference in gene regulation, but only in one specific population group.
Exactly. And since ZBTB7A is known to be a repressor of glycolysis, how cells use sugar, and also fetal globin, this finding could have really intriguing specific implications for metabolism or maybe even blood disorders that differ between sexes uniquely in that population.
This naturally leads us to a really critical area, population-specific eQTLs. Now, for our listeners, can you break down what exactly eQTLs are and why they're so important in this context?
Sure. eQTLs stands for Expression Quantitative Trait Loci. It sounds complicated, but the concept is fairly simple. They are just genetic variants, small differences in our DNA sequence that influence the expression levels of a specific gene.
Okay.
Think of them like volume knobs or dimmer switches for our genes. A slight genetic variation at an eQTL site can turn a gene's activity up or down in certain cells.
Got it. Like dimmer switches. And AIDA found a lot of these.
They did. They identified over 11,000 unique genes that had at least one cis eQTL. Cis just means the variant is located close to the gene it controls on the DNA strand.
Okay, 11,000 genes with these regulatory switches nearby. But what's the big takeaway about these eQTLs when we look across diverse populations, like in AIDA?
Yeah, this is really the crucial insight. A significant proportion, nearly 7% of all the eQTLs identified in this diverse Asian cohort were found to be low frequency or even rare in non-Asian populations, like African, admixed American, and European groups, based on existing databases.
7% were rare or low frequency elsewhere.
Yes, and even more strikingly, about 2.24% of these AIDA eQTLs were entirely absent in European populations. They just don't have that specific genetic variation.
Ow.
And when they compared their findings directly with the GTEx project, which, remember, is predominantly European-based data, over 20% of the eQTLs that were only identified in AIDA, not found or significant in GTEx, involves a genetic variant that had a low minor allele frequency in Europeans.
So 1/5 of the new eQTLs they found were linked to variants uncommon in Europeans.
Exactly. It definitively demonstrates the sheer abundance of population-specific genetic variants out there that are actively impacting gene expression. These aren't just silent differences, they're functional.
That's truly astounding. It means if our reference data sets, the ones we use for drug development and understanding disease, are based only on European populations, we're fundamentally missing a massive chunk of the genetic landscape that controls gene activity in other groups.
That's precisely the issue.
And they also found that some of these eQTL effects were context dependent, right? What does that mean?
Yes, further complicating the picture, but also making it more biologically realistic. Some eQTL effects weren't constant. They actually varied based on the cell's context or its current state.
Like what?
For example, they looked at a variant called RS7239151. Its impact on the expression of a gene called CYB5A actually varied depending on the B cell's activation status, which they measured using something called a blue module score related to antibody production. So the same genetic variant had a different effect on gene expression depending on what the B cell was actually doing biologically at that moment.
So genetic effects aren't always static. They can be modulated by the cell's environment or activity.
Exactly. It adds another layer of intricacy to understanding how our genes influence our biology.
So putting this together, if the genetic switches, these eQTLs that control gene activity, differ so much across populations, and their effects can even change depending on what cells are doing, what does that really mean for how we should be thinking about developing and using things like gene therapies or even just targeted drugs in the future? It raises some truly fundamental questions for personalized medicine.
It absolutely does. You need to know which switches exist and how they work in the specific population you're treating.
Okay, now let's connect all these molecular dots. We've got different cell proportions, different gene expression, population-specific eQTLs. How does this actually link to disease risk? AIDA used colocalization analyses for this, right? Can you explain that?
Yes. Colocalization is a statistical method used to determine if two different signals, in this case, an eQTL signal, a genetic variant affecting gene expression, and a disease risk signal from a genome-wide association study are likely driven by the same underlying causal genetic variant.
So it's like finding where the gene's volume knob and a known disease risk factor line up exactly on the genetic map.
That's a good way to put it. It helps pinpoint which specific genetic variants, acting through their effect on gene expression in specific immune cells, might actually be driving the disease risk observed in GWAS studies, especially when looking within specific populations like the Asian cohorts used here.
And did they find compelling examples of these population-specific links?
They did, providing numerous examples where variants more common or functional in Asian populations are linked to immune-related diseases. For asthma, for instance, they highlighted a variant called RS5763110.
Okay.
This variant acts as an eQTL for a gene called SMA2, specifically in CD4 plus T memory cells. And importantly, this same variant is a known GWAS hit for asthma risk. Now, this specific variant is relatively common in Asian populations, but has a low frequency in African and European groups. And we know that the biological pathway involved, TGF-beta SMAD2 signaling, is very active in the airways of people with asthma. So this provides a specific molecular link, potentially contributing more significantly to asthma risk in Asian populations.
That's a really clear example of how these underlying genetic predispositions can vary geographically, directly impacting who might be more susceptible to certain conditions based on these population-specific variants. What about other diseases?
They found similar connections for rheumatoid arthritis, or RA. A variant, RS223-0500, was found to be an eQTL for the HIF1A gene in a specific type of B cell, IgHM low memory B cells.
HIF1A involved in inflammation.
Exactly. And this same variant was linked to RA risk in both trans-ancestry and East Asian Jedi-Gordy's studies. But again, this specific risk variant is rare in non-Asian populations. Another really interesting one for Graves' disease, an autoimmune thyroid condition. They found a variant, RS74416-240, acting as an eQTL for a gene called TCHP in CD14+ monocytes. This eQTL signal was identified only in the East Asian superpopulation data within AIDA and was linked to Graves' disease risk.
And is TCHP well understood?
Actually, very little is known about TCHP's specific role in disease. So this finding, directly enabled by studying this diverse cohort, flags TCHP as a strong candidate gene for future research into the mechanisms of Graves' disease, particularly in East Asian individuals.
It really shows how studying diverse populations can uncover entirely new biological leads, even for something as commonly measured as, say, lymphocyte counts in a blood test.
Exactly. They found a variant, RS1794188, that acts as an eQTL for a gene called Ceslint1L in CD16 plus NK cells. This variant is also a known GWAS hit for lymphocyte counts.
And the population difference?
Again, this variant is rare in African and European populations, but common in East Asians. And Ceslint itself is known to be important for the function of another immune cell type, CD8 plus T cells. So these aren't just abstract statistical correlations. They consistently point towards specific biological mechanisms potentially operating differently or at different frequencies across populations.
So after hearing all this incredibly detailed evidence, what does it all really mean for the bigger picture of medicine and health? The so what here feels immense.
The so what is, I think, incredibly clear and quite profound. These findings powerfully underline both the scientific necessity and, frankly, the ethical imperative of studying diverse populations.
Scientific and ethical. Okay.
Yes. It's not simply about having a more complete, more accurate picture of human biology, although that in itself is a massive scientific gain. It's fundamentally about enabling personalized medical care that is actually effective for everyone. It's about achieving truly equitable health outcomes globally.
Because the alternative is?
The alternative, if we continue to base our medical understanding, our diagnostic tools, our treatments, primarily on data derived from an unrepresentative segment of humanity, is that we will inevitably perpetuate and likely even exacerbate health disparities for the rest of the world. That's the real danger.
That's a powerful statement. Now, while the AIDA study is clearly incredibly rigorous and groundbreaking, it's also important, as with any scientific research, to acknowledge potential limitations, right?
Absolutely. Science is always an ongoing process. One key thing to remember is that the associations we observe between self-reported ethnicity or genetic ancestry and these cellular traits are complex.
How so?
Well, they likely represent a combination of both genetic factors, which we've discussed a lot, and environmental influences, things like diet, geography, exposure to different pathogens, lifestyle factors. And these genetic and environmental factors are often confounded, meaning they travel together in real-world populations, making it challenging to entirely disentangle their individual effects just from this data alone.
So genetics plays a role, but environment is intertwined.
Exactly. Future studies will ideally need even more comprehensive metadata, detailed information on these environmental and lifestyle factors for each donor to really start teasing apart those relative contributions more effectively.
Any other limitations?
Well, as careful as they were with harmonization, the authors do acknowledge that despite their meticulous methods, some small amount of residual technical variation across the different international study sites might still exist. It's incredibly hard to eliminate entirely in such large multi-center studies.
Understood. Important caveats. So maybe let's try to put AIDA into perspective with a final summary. Disease links. AIDA clearly shows how population-specific genetic variants, these eQTLs, impact disease susceptibility for conditions like asthma, rheumatoid arthritis, and Graves' disease, often through mechanisms that might be more prevalent in Asian populations. Future diagnostics. It strongly highlights how single-cell genomics is really paving the way for the next generation of diagnostics. You could imagine a highly advanced, super detailed CBC blood test in the future that gives you a complete immune blueprint, perhaps even tailored to your specific ancestral background.
A deep CBC,
A
perhaps.
deep CBC, I like that. And global health.
Yeah.
Crucially, the overarching message is that the inclusion of diverse populations in these foundational biological atlases is absolutely paramount if we want to achieve true global health benefit and equity.
Couldn't agree more.
So we've seen through this deep dive into the Asian Immune Diversity Atlas just how profoundly human diversity impacts our immune system at really every single level, from the
Yeah.
proportions of different cell types right down to the expression of individual genes and their complex regulatory networks.
It's woven into our biology.
And ignoring this incredible diversity means we're constantly missing crucial pieces of the puzzle for understanding, diagnosing, and ultimately treating disease effectively for everyone.
It's become very clear that a one-size-fits-all approach to medicine, especially if that one size is based on historically limited population data, risks leaving vast segments of humanity behind. And that's a scientific reality I think we can no longer afford to overlook.
So here's a thought for you, our listener, to carry forward from today's discussion. If a common medicine you might take, or a widely used diagnostic test, was developed primarily using data from one specific population group, how might it unintentionally fail or perhaps disadvantage someone from a different background?
It's a critical question.
And maybe more personally, what specific steps can you, as an informed individual, take to advocate for more diverse representation in scientific research?
Yeah.
How can we all help ensure its benefits actually reach everyone, everywhere?
And building on that, what new questions does this deep dive raise for you about the future of personalized medicine and that larger, absolutely critical picture of global health equity?
Lots to think about. Thank you so much for joining us on this deep dive into the truly fascinating and vital world of human immune diversity. Keep learning, keep questioning, and keep exploring.